Okay. Good afternoon, one and all. So, in morning lecture only, we have discussed that what is the meaning of the word isotonic solutions. What happens if the solution is not isotonic? That we are going to discuss today. So, what is the meaning of the word isotonicity? What is the meaning of the word osmotic pressure? That we have discussed in detail in morning lecture only. What is the meaning of the word osmotic pressure? It is the minimum pressure which is exerted by this semi-permeable membrane or the pressure applied to a solution to prevent the inward flow of the water across the semi-permeable membrane. So it is defined as the osmotic pressure. Then what is the meaning of the word tonicity? It is the ability of the extracellular solution. So this is an ability of this extracellular solution to make water to move in or out of the cell. So this is nothing but the tonicity. So in order to avoid the any uh, disruptions to the cells or the semi-permeable membrane, the solution must be isotonic with that semi-permeable membrane. So what is the meaning of the word isotonic solution? It refers to the when two solutions have same osmotic pressures. So whenever the two solutions have same osmotic pressure, that solution is known as isotonic solutions. So that example of this isotonic solution we have discussed already. 0.9% sodium chloride solution is known as isotonic solution. It is also known as physiological saline solution or a normal saline solution. This we have discussed in morning class only. Now we are interested in the terminologies, that is hypertonic solutions and hypotonic solutions. So what is the meaning of the word hypertonic solution? So it is nothing but a tonicity is more than the RBC. A hypotonic solution is one that has a greater concentration than the reference solution. That is RBC's contents. That means the concentration or the solution's concentration is more than the RBC's tonicity. Kiva RBC's osmotic pressure. A hypertonic solution has a greater osmotic pressure than that of the reference solution. And if we are going to take an example, that if we are going to suspend RBCs in 2% W by B solution of sodium chloride solution. See, we know 0.9% percentage of sodium chloride solution is isotonic with the RBCs. Now, we are exceeding here the concentration of sodium chloride solution, that is about 2%. And if we are suspending these RBCs in the 2% solution of sodium chloride, then the water present within the RBCs will come out. To dissolve this or to dissolve this 2%, as the osmotic pressure inside the cell is minimum as compared to the outside extracellular fluid, that is 2%. So the water which is there in the RBCs will came out of the RBCs. The water which is present in the RBCs will came out due to the osmosis from the dilute RBC fluid to the concentrated hypertonic solution, because the extracellular fluid has a more concentration than the RBC cell membrane. So water which is there in the RBCs will came out. That's why the RBCs will shrink out. So the exist of the RBCs from the RBCs causes their shrinkage and RBCs become wrinkled in the shape. That's why these RBCs will be wrinkled, because this black is nothing but the extracellular fluid having a concentration of 2% W by B solution. So the water which is there in the RBC will came out to dissolve the solute which is there in the extracellular fluid. As water came out from these fluid to extracellular, these RBCs will shrink out, and the shrinkage of these RBCs, this phenomenon is known as plasmolysis. That means a hypertonic solution will do the plasmolysis of RBCs. What's the reason behind that? As extracellular fluid contain 2% concentration of sodium chloride solution, and the osmotic pressure is less in the RBC cell. So what is water which is there in the RBC will came out to dissolve the solute molecule which is there in the extracellular fluid. As water came out of the cell, RBCs will shrink out, and this shrinkage of RBCs is known as plasmolysis. So this is with respect to the hypertonic solutions. When we are going to discuss about hypotonic solution, if the tonicity is less, a hypotonic solution is one that has a lower concentration than the reference substances. See here, we are going to take a hypotonic solution, that means 0.1% solution of sodium chloride. See, 0.9% is isotonic, 2% is hypertonic. That means 0.1% is 0.1% is hypotonic. See, if hypotonic solution is having a low concentration than the reference solution, so what happens? Has a lower osmotic pressure than the reference substance. So RBCs, if we are going to suspend in a 0.1% solution of sodium chloride, then water from this solution will enter into the RBCs. As the osmotic pressure in the RBC is less than the extracellular fluid. So the RBCs, so the water which is there in the extracellular fluid will come inside the RBCs to maintain the same osmotic pressures outside and inside. That's why the water which is there in the extracellular fluid will enter into the RBC cell. Due to the entry of water inside the RBC cell, RBC will swell out. See, RBCs are suspended in 0.1% solution of an ACl. Then water from this solution will enter into the RBCs. And due to the entry of water inside the RBCs, causing them swell, which may result in rupturing of the RBC. See, here this picture will depict us that the rupturing of the RBCs will happening due to extra amount of water has entered into the RBC cell. RBC will swell up and they will rupture this RBC. So the rupturing of the RBC will release the hemoglobin. See, if RBCs will rupture, what are the contents of RBCs? It is an red blood corpuscles. So whatever hemoglobin inside the cell which will came out of that RBC cell. So this process of rupturing of the RBCs and leakage out of hemoglobin outside that RBC cell, this process is known as hemolysis. That means hemoglobin has came out of the cell of RBC. That's why this process is known as hemolysis. That means isotonic solution will know, see here, we are going to do the comparative study of all those three solutions: hypertonic, isotonic and hypotonic. Isotonic solution, if it is required, then water came inside. If it is not required, then water is sent outside. That's why the reason behind that, it is going to have the same osmotic pressure with that of that outside the membrane. In case of hypertonic solutions, if we are going to suspend the RBCs in the hypertonic solutions, so water which is there in the RBC will be entered in the extracellular fluid. That's why the shrinkage of the RBC will happen. And with respect to the hypotonic solutions, as the tonicity in the RBC is less as compared to the extracellular fluid, so water which is there in the surrounding fluid will go inside the RBC. So RBC will swell up, and here the RBC will be shrinkage out. So in case of hypertonic solutions, plasmolysis will happen, and in case of hypotonic solutions, hemolysis will happen. But there is no any effect with respect to the isotonic solution, as it is totally compatible with the red blood cells. So in this way, the effects of hypertonic, isotonic and hypotonic solutions are there on the red blood cells. So there is a very important point to measure the tonicity of all those pharmaceutical preparations must be there. When the pharmaceutical preparations are of ophthalmic use, when the pharmaceutical preparations are of blood compatible use. So we have to measure the tonicity of these formulations. So the tonicity of the solution may be determined by given following methods. Now what are those following methods? Method number 1 is hemolytic method and method number 2 is colligative method. So what is the meaning of the word tonicity? It is the measure of relative concentration of solute particles on either side of the membrane, that is outside the that is in the extracellular fluid or in the intracellular fluid. Only solute that cannot cross the membrane contribute to the tonicity. See which type of compounds or which type of particles will affect the tonicity. The solute particles which cannot cross the membrane, those only particles uh, will give or will contribute to the tonicity. It determines the direction and extent of which water moves through the osmosis. So in case of hypertonic, the water moves from intracellular to the extracellular, but in case of hypotonic, water moves from extracellular to the intracellular. So this is totally depending upon the concentration of that extracellular fluid, whether it is also isotonic, whether it is hypotonic, or whether it is hypertonic. So there is need to determine the tonicity of the, or there is need to measure the tonicity of these formulations. So there are two very important methods to measure the tonicity. Number one will be hemolytic method and second will be colligative methods. So what is the meaning of the word hemolytic method? Now, the hemolytic method, in this method, the effect of various solutions of the drug is observed on the appearance of red blood cells when they are suspended in the solution. See, just now only we have seen the comparative study of three solutions: hypotonic, isotonic, and hypertonic. So when we are going to do the comparison of this, so this is very important parameter to measure the tonicity. If there is no change in the size and shape of the RBCs when immersed in that test solution, if RBC have no any change, if it is suspended in any solution, that means that solution is isotonic with that RBC cells. So whenever we are going to view or whenever we are going to observe these under the microscope, then we can measure whether the solution is isotonic, whether the solution is hypotonic, or whether the solution is hypertonic. So this method can be made more accurate by using hematocrit. So the measuring of the size of RBC and appearance of the RBC. For this, if we are going to use hematocrit, it is very easy method, which is a centrifuge method in which a graduated capillary tube is held in each of the two arms. See here, what procedure we are going to follow for the determination of tonicity in the case of hemolysis method. See, the procedure for this is one capillary tube, that is tube A, is filled with a blood which is diluted with 5 ml of 0.9% sodium chloride solution. See, in case of tube number A, we are going to fill blood which is diluted with 5 ml of 0.9% sodium chloride solution. And the other capillary tube, that is tube B, is filled with a blood diluted with equal volume of 5 ml of test solution. See, in one tube we are going to take blood with the isotonic solution, that is 0.9% sodium chloride solution, and in another tube we are going to take blood with a test solution. Now we are going to centrifuge both the tubes, that is they are going to rotate it at high speed at a centrifuge machine. After centrifugation, the blood cells are concentrated at the end of the capillary tube and the volume occupied by the cell, that is packed cell volume, is measured. So after centrifugation, see here, we are going to withdraw the blood and it is diluted with 5 ml of either isotonic solution or the test solution. Then both the tube we are going to centrifuge at high speed, they are rotated in this machine, and uh, after that, after centrifugation, the blood cells are concentrated at the end of the capillary. Then RBCs will settle down at the bottom of the tube. So the capillary tube, and the volume occupied by this packed cell is measured. After centrifugation, the volume which is occupied by this packed cell is measured. So finally, the packed cell volume of the test solution is compared with that of the isotonic test solution. After centrifugation, we are going to compare both the glass tubes, one which is produced by the test solution and another one which is produced by the isotonic solution. So we are going to do the comparison of both these tubes, and we are going to obtain the interferences of that. See, so in this way the tube will look like. The blood without RBCs will be plasma, or the solution without RBC is plasma. Then in between these two layers there is a coat, that is a buffy coat, which contain leukocytes and platelets, and a bottom of the tube, that is there is sedimentation of the RBCs will happen, which is 45% of the whole blood. So what will be the result of this comparison study or conclusion of our result? So the packed cell volume of the test solution is same that of their test A. If packed cell volume of both the test tubes or the both capillary tubes are same, then our solution is isotonic. If the pack volume of the tube is more than that of the test, if the pack volume of the test is more than that of this isotonic solution, then our solution is hypotonic, uh, because the RBCs will sell out and they will increase the pack cell volume, which occurs in the case of hypotonic solutions. Now, in case of third case, when if the packed cell volume is lower than that of the isotonic solution, if it is less than that of tube A, then the test solution is hypertonic solution, because there is decreasing in the packed cell volume than that of the uh, isotonic solution. So this is method number 1 for the determination of uh, isotonicity. So there will be a question that they write a note down, not on measurement of tonicity. So the method which we have discussed now is very important method for the determination of tonicity. There are another 4 different methods also. These methods are known as colligative methods. Now this colligative method it is there in your syllabus also, but these methods you can see in the pharmaceutical syllabus in depth. Just we will revise them here. So this colligative method, it has been determined that solutions having same tonicity exhibit similar behavior with respect to the colligative properties. Now these colligative properties are those properties which are similar to the isotonic solutions. Now what are those properties basically? Properties such as lowering of the vapor pressure, depression in the freezing point, tonicity of the solution may be determined by determining the colligative properties. Colligative properties means those properties which are same with the isotonic solutions. So what are those 4 different properties? For making the isotonic solutions, the quantity of the substances to be added may be calculated by following 4 methods. But for methods, that is colligative methods. And what are those colligative methods? First method is based upon the determination of molecular concentration. Second method by depending upon the freezing point data. Third method is based upon the sodium chloride equivalent volume, and fourth method is depending upon the White-Vincent method. So these 4 methods are there for the determination of uh, isotonicity, or for the determination of tonicity of the given solutions. So we'll just discuss the principles of all those 4 methods. First method from the colligative property is based upon the molecular concentration. If 1 gram of molecule of a substance is dissolved in 100 ml of water, the resulting solution will be 1%. We know if we have 1 gram we are going to dissolve in 100 ml, then the solution is of 1%. So for example, the molecular weight of boric acid is 62. So if 62 grams of the molecular weight of boric acid is dissolved in 100 ml of water, the resulting solution will have 1% molecular concentration. So for non-ionizing substance, an aqueous solution having 1% molecular concentration depresses the freezing point to 18.6 degrees centigrade. And freezing point of the plasma is minus 52 degrees centigrade. Um, it is 0.52 degrees centigrade. So by using this information, we can calculate the molecular concentration of the blood plasma IFLO. A depression of minus 18.6 degree freezing point of solution is due to the 1% molecular concentration. So all these data you can study in detail in the pharmaceutics syllabus, because calculations are there for you in the pharmaceutics. So this is today what I am going to deliver, that is uh, calculation of tonicity, from which only one method is important, that is hemolysis method.